Welcome to part three. So now we're going to review the heart and the heart tissue from the microscopic view. And this is going to be important because we're going to learn about the electricity in the heart, and we're going to talk about AKG. You don't need to know all the details about AKG, but the pure basic stuff. So it's important to recognize how the tissues, specifically the heart, from the um, microscopic level, how they work, right? First, we need to know two things about the heart cells. So there are two types of heart cells. The first type is called the pacemaker cell. Here, these pacemaker cells are specialized cells that they look almost like neurons. They generate action potentials, which, if you remember from AMP1, they're like electrical impulses, kind of, sort of. And those are going to be, as the name implies, pacemaker cells are the ones that are going to dictate the velocity, how the cells are going to contract, and how the heart rhythm and the heartbeat will be, right? So the pacemaker cells, all they do is to generate electrical or action potentials. To what? To stimulate the myocardial Okay, so myocardiocytes, those are the cells that contract. Those are the cells that we can see here. And those cells, if you remember, when we learn about the muscle tissue, all your tissues, they need nerves, or they need some sort of electrical impulse so the contraction can take place. And we said the heart is affected by nerves. So you have a group of nerves that will increase the velocity of the heart. You have a group of nerves that will directly stimulate the heart to reduce the heart rate. But if you remove that part of the nervous system, if you remove the nerves, in the heart itself, you have these pacemaker cells that auto-generate electrical impulses and stimulate the myocardiocytes to contract. That is what this is about. The autorhythmicity is the ability to initiate an electrical potential, or in this case, an action potential, that will trigger muscle contraction. That is one of the features of the heart. What else is important about the heart? That the way how the heart cells, the myocardial cells, are connected to each other is pretty important, because if you look at as in general, the whole idea is that each individual cell, they need to contract synchronically with the other one. So what I'm saying is all these cells need to work as a team to contract at the same time, so the whole organ will contract in a synchronized way, right? This is able, or this happens because the cells are interconnected through these regions called intercalated discs. Okay, this intercalated disc, if we look at this uh, picture here, you can see this open down thing. What I'm covering here or showing you here is a single muscle cell. So the muscle cells are short, they have these kind of branches. And if you look at that space even closer, let's say that this is the open down thing. Let's say that this is the cell number one, and this is another cell, so cell number two. There's these gap junctions, which essentially are little tubes that connect these two cells together. I mean, there's desmosomes. They're like glue. They're like sealed, and they keep the cells together. But the thing is that if you have an electrical impulse, an action potential, the action potential will not be able to reach the second cell unless you have a gap junction. Okay, so that's why these gap junctions are important. That's why these intercalated discs are important. because if a pacemaker cell stimulates a single myocardiocite, because of that connection, the whole muscle will respond at the same time and will contract as a unit. All right, remember what I said about keeping the mindset that this thing is not complex and you can understand that. I'm saying this because the students, especially in the lab, confuse this all the time and it gets like frustrated and everything, when again it's not a big deal. It's really simple. I told you that we have those pacemaker cells, right? Now, when you have a bunch of pacemaker cells together, there's going to be something called nodes. So we have basically two nodes, and again, two nodes is a bunch of pacemaker cells together. Remember, the pacemaker cells are the ones that generate uh, electrical impulses. So the key here is to look for the right atrium, because in the right atrium is the place where you're going to find the two nodes. If you look at the picture here, we have the superior vena cava, and right in the most superior part, this little node that is here in yellow, again, don't get familiar with colors or textures, basic shape, right? A circular structure there is your SA node. The most important one, the SA node, is also known as the pacemaker of the heart. This part that it says the SA node has the highest inherent rate of depolarization is a fancy way to say that this SA node is the one that fires first or faster than the rest of the system. So, and then we have another second node, which we're going to review here. The AV node or atrioventricular node is still located within the right atrium, but it's a little bit lower. So it's almost close to the atrioventricular valve, as you can see here. Okay, so those are the two nodes. Those are going to be very important when we learn AKG. When you learn action potential, we'll see how they work. All right, let's review these quickly, and it's better if we just look at the pictures. All right, so we have the superior vena cava, we have the inferior vena cava. This is, of course, the right atrium, right ventricle, left atrium, left ventricle. This in the top is the SA node, and this one in the bottom is the AV node. You have to use imagination here, and this follows the same principle as we're going to see in terms of, if you see the AV node is just this circle over here, this area, okay? Now notice that from the AV node, then we have this little string or this little bundle of things. I don't want to call neurons or nerves, but we have this bundle that eventually. So what I'm saying is, do you think that that little thing is still called the AV node? No, node. Node. Do you think this is called the same? Of course not, because it's not a node, right? It's going to have a different name. And when that specific structure then splits in two, the name will change. This will be left and this will be right, blah, blah, blah. And then eventually, when that bundle branch reaches the tip or the apex and start turning towards the ventricles, the name also will change. So I'm saying that right now just to have, so you can keep that in mind. But as you're going to see, the name of these structures of this conduction system will change. So step number one is that the SA node fires. So in other words, release an action potential that is represented here by these arrows. Okay, so and notice that there's one that crosses and reaches the left atrium. So what this electricity is spreading through the muscles of the atria, And also one of them is going to stimulate the AV node. Now the AV node, there is a little bit of pause that it takes a fractions of seconds to completely stimulate the AV node. What I'm saying is when the action potential with the SA node fires, that electricity passes extremely fast and quickly, and that leads to that contraction, what we call atrial systole or contraction. And then the term that you need to get familiar with when a cell depolarized, when the action potential reach, that is what we call depolarization. So activation of the SA node will do two things. Will depolarize all the myocardiocytes in the right and left atrium, and at the same time is going to activate the AV node. But the AV node, it takes a little bit of time before that the action potential is spread to the rest. So that's what we're going to see after. So that little pause is pretty important because that pause allows the atrial cardiomyocytes, which they're also known as myocardiocytes, cardiomyocytes, the same thing. Um, so that pause allows these cells to contract before, and that is the key word here, before the ventricles. And now we know, hopefully you remember why this is important, that the atria contracts first and then the ventricles after. Once the AV node is stimulated, notice that the atrial contraction already passed, then the AV node extends or continues with this structure that I have here. Is called the AV bundle. So atrioventricular bundle. So it's what continues after. And so once again, the little ball, the little circle is the AV node. This single cord is the atrioventricular bundle. And notice that why is important the interventricular septum, right? Because through this muscle, through the atrioventricular septum, in other words, if you cut or if you dissect the interventricular septum, you're going to find those bundles and bundle branches, okay? So the AV node leads to the AB bundle, and the AB bundle is called that way when it's just one. See what happens after, or where you can see over here. When it splits in two, it's no longer called atrioventricular bundle. It's called either left bundle branch and right bundle branch. So, and I mean, I think it's pretty simple, but you'll get surprised how many people sometimes do not get that straight. That's why I'm repeating this. A, V node, and you have a single one. This is what we call the bundle. And then when the bundle is split, that is going to be the right bundle branch, and this one's going to be the left bundle branch. And they're going to be called bundle branches until they reach the apex of the heart. Once they reach the apex of the heart and turn to expand through the ventricular wall, right, they're going to be called Purkinje fibers. So, in other words, from here to here, all of these is what we call the Purkinje fibers. And you know, this happens extremely fast. Once the AV node fires, the bundle branches and all of these electrical impulses will travel extremely fast. And then that action potential is what stimulates the cardiomyocytes. And the ventricular systole or depolarization will take. Okay? So that is the conclusion is that at the end contraction occurs, and then after that, what comes after contraction? Then relaxation, and as you see, the cycle repeats between systole and contraction. Now, Why this is important? Because when we learn the AKG, we're going to see in the AKG where the electricity is running in specific phases of the cardiac cycle. In terms of the membrane potentials and the action potentials and depolarization, repolarization, this is a concept that we learned in AMP1. We're going to mention very quickly. When the cell membrane, when the cells are at rest, they have a negative voltage. Okay, so an action potential is a change in the voltage inside of the cells. And the most important elements or ions are sodium, calcium, and potassium. And notice that all of them, they have positive charge, right? So important here is to recognize that depolarization is the influx of sodium. So depolarization means that in the cell, all the voltage channels that are specific for sodium will open, allowing an influx of sodium to getting inside of the cell. And because sodium has a positive charge, and of course sodium is, we have more sodium outside of the cell than inside of the cell by concentration gradient, the sodium gets inside of the cell. And as all the channels open at once, sodium start getting inside of the cell, and that the cell start changing from negative to positive. Okay, in some point, that positivity, that change in voltage, is what we call an action potential. And that opens calcium channels that will keep this this part of the action potential called the plateau here less important for your exams, and I don't want to be specific. But what I think is important to recognize is that depolarization represents the influx of sodium ions getting inside of the cell. Okay, when we, and this is what the AKG will detect, that change in voltage. Okay, so this is what happens when the SA node activates, and this action potential then spread. Now, after depolarization, we have another phenomenon called repolarization. So repolarization is when potassium exits the cell. Potassium, we have more potassium inside of the cell than what we have outside. So once you reach a voltage of approximately plus 30, that is the voltage that opens potassium channels. And all the channels, once they open, they let an exflux or an exiting of a large amount of potassium. Potassium has a positive charge, so that means that the cell will lose positivity and will become again negative, right? So that is depolarization. If you have a headache already with all this nonsense that I just explained, let me just summarize this for your exams. The most important thing is to be able to recognize sodium and potassium. Repolarization, okay? Think about re as rest. So when the cell is resting, meaning it's not stimulated, when the cell is not contracting, this is what we call repolarization, and it has to do with potassium leaving the cell. Therefore, the cell becomes more negative because potassium has a positive charge. Depolarization is the opposite. So depolarization, what is the opposite of resting? Being our action, or in this case, meaning an action potential. So depolarization is the changes in voltage inside of the cell that is due to the influx of sodium, which has a positive charge inside of the cell. With all that information, now we can talk about electrocardiogram, an AKG, all right, for sure. Now, this is something that you have seen probably in movies or in emergency room, that whenever somebody has a heart attack or whatever, they do an AKG, and they have this AKG is a little machine, right? Has a bunch of buttons here, has um, a screen here with a monitor, and then it has like a little printer which is going to print the AKG. This machine has a bunch of cables that connect the specific parts of the body. You have one of you put here in the wrist, you put one here in the lower part, and then you have all these numbers: B1, B2, B3, B4, B blah blah blah, B6 in total. um, those are called electrodes. That doesn't, at this point, you don't need to know the details about that. You're going to learn about how to place those specific electrodes in specific areas to measure the electrical activity of the heart. Okay, that is the key. And what you need to know for your exams or is the most basic part of the AKG, which is to be able to identify these P wave, QRS complex, and T waves. Okay. Remember that we said depolarization is when you have an influx of positive charge, right? So a neutral or at rest, you have like a line. If you have a depolarization, this will go up because positive charge are going, more, and this is what the AKG will, these electrodes will pick. When you have repolarization, we have an uh, we have positive charges leaving the cell, so the cell become more negative, right? So what that means is you're going to have a deflection, or the AKG will go towards the bottom, right? So at the end, when you have this is what we call a wave, and we have a P wave, we have a big old wave called the QRS complex, and then we have a T wave, and those waves represent depolarization of a specific parts of the heart, which I'm going to show you now what does that mean. All right, look, if you memorize this, that's all what you need to know. First, P wave. What is the P wave? What does that represent? The P wave represent the electricity that the electrocardiogram will pick when the atria is depolarizing, meaning the SA node fires. Remember the action potential? The action potential spreads. That is depolarization. And we know that after that, what follows is atrial contraction. So in the AKG, the P wave is this first portion. Now notice that then we have like a little pause, right, between the P wave and this QRS complex. We have a little pause. I'll show you what does that pause means. But the QRX complex, notice what that represents. So the QR complex represents the depolarization of the ventricles, and you can see that the positivity is way higher than the P wave, just because remember that ventricles are larger and bigger, so there's more cells and there are more amount of electricity running through them, right? So that is the depolarization of the ventricles. And then when the cell depolarizes, that means that the cell needs to repolarize in some point. The T wave, which is this one that we can see here, will represent a repolarization, right, of the ventricles. So you might be wondering, where is the repolarization of the atria? And if you read here, we'll tell you that the repolarization of the atria in the AKG, we cannot see it because it's masked with the QR complex. So in other words, if we were able to see it, the repolarization of the atria will be somewhere here, but we cannot see it because the big QR complex basically covers or erases, so we cannot see that one. Right, so everything starts with the SA node. The SA node fires, and that is what generates the P wave, depolarization of the atria. Fractions of a second after that, that causes contractions of the atria. At the same time, while the muscle is contracting, notice that there's this little pause. That is the time that it takes to stimulate the AV node, so the AV node can fire and um, conduct or transmit the electrical impulse to the ventricles. Also, it's important to recognize here, and this might be extra, you might be wondering why the electrical impulses don't go this way, right? It's because the valves that we have here, the atrioventricular valves, are a matter of cartilage and material that do not transmit the electricity. They're like some sort of a isolation, so the electrical impulse cannot pass through the valve. So that's why the only way how the electrical impulse will reach the ventricles is through the AV node. After the pulse, then the AV node fires, and then the electrical impulse passes almost instantly and is spread through the whole bundle branches, and that is what generates the QR complex. We know that now that the impulses reach the muscles and the ventricles, right immediately after that, what you're going to have is the ventricular contraction, right? And then the ventricular contraction, you have a pause because that's the time that it takes to the ventricles to fully contract. And then eventually when the ventricles now relax, that relaxation is what we call the T wave and describe repolarization of the ventricles. The QR complex, depolarization of the ventricles. The P wave is depolarization of the atria. Okay. The concepts about the cardiac cycle, I think we will cover this very quickly because we've been talking about this a lot and it's just pure repetition. So we're going to see the AKG, we're going to see what happens with the contractions, and of course we need to see how is the blood flow, what happens with the valves, because all this is now basically putting all of those concepts together so you will have the whole picture of what's going on with the heart. Remember that a cardiac cycle is a period of time that begins with contractions of the atria and ends with ventricular relaxation, right? We said contractions, systole, and relaxation, diastole. Okay, let's start the cycle with both atria and ventricles relaxed, okay? So basically that means atrial diastole and ventricular diastole. This is that period of time when blood flows to fill the ventricles with blood, right? Just by that relaxation, I told you before, about 70 to 80% of the ventricular filling occurs by this method. Okay, of course, the tricuspid valve and the mitral valve in this specific period of time, they need to be open, both of them, to allow that blood to go from the atrium to the ventricle, respectively. The blood reach about 70%, right? Then is when we have the atrial contraction, or sometimes it's referred as the atrial kick. This is important because contributes to the remaining of the 20 to 30% of the filling of the heart, as I mentioned before. In this case, in that specific moment, we have an atrial systole contraction, right? If we associate with what just happened electrically, remember that the SA node at this specific moment or a few fractions of seconds before just fired the action potential, right? So the SA node fires the action potential reaches all the walls of the atria, and then the atria contracts. Remember, the AV node is taking a pause, so the action potential is not reaching the ventricle. That's why the ventricles are still in diastole, and that's why important. That's why it's important, that pause. And so yeah, that's what that means. Atrial systole. That's why it's important that both atria contract right at the end to completely, or to completely fill the ventricles with blood, 100%. Okay, what is next? The ventricles are full. Now we need to squeeze them, right? Now, let's keep this simple. There's a little bit details about the ventricular systole, phase 1, phase 2, isovolumetric, blah, blah, blah. Don't worry too much about it in terms of the exams. You might have questions about these in the quizzes, but not too much for the exams. And you will see, what does that mean? Okay, so we have, now that the ventricles, that the atria has complete the systole, then the atria is in diastole, as you can see here, right? And then the ventricles start systole, contracting. So, and this is extremely fast, right? Just fractions of seconds. There's a point which is called the isovolumic contractions, in which the ventricles are contracting, they start contracting, right? Is this contraction generates enough pressure to close the tricuspid and mitral valve, but the pressure has not reached the point enough that is required to open the pulmonary aortic valve. So in other words, you can see here that everything is closed. So basically there's not blood flow yet. And that's why this is called isovolumic contraction. So blood is not being ejected yet. And again, this is very, very little, because as you will see, really, the ventricles will keep contracting, and then will reach a pressure that will be high enough to open these semilunar valves, and then ejection will take place, which is what we can see here. This phase 2 of the ventricular systole. Now this is called the ventricular ejection phase. I'm describing this just for your quizzes. Don't get too crazy with this for the exams. It's not worth it. But what you need to know is that during ventricular systole, this is the specific time when the QRS complex, we can see it in the AKG, and that's the time when these valves open and allow the blood flow. And I like to show you this one because you can see that the right ventricle and the left ventricle, they're contracting at the same time, and these valves are opening at the same time, and then the tricuspid and mitral valve, they close at the same time. And that's why this two, when they close at the same time, they generate the sound S1. What else we need to know here? Okay, so when this blood gets ejected, right, the amount of blood that is ejected with one contraction is what we call a stroke volume, right? The stroke volume is on average at rest about 70 to 80 milliliters of blood. That is how much blood you normally, or on an average person, will eject with a single contraction. Now, this stroke volume, it can change, of course. There's many factors that can affect. For example, how strong is the contraction? That will increase, the stroke volume, or how much blood the ventricle have before contraction. That will also determine the stroke volume. Those are details that you don't need to know, but just know, and I'm just mentioning stroke volume because this is important for your exams. The stroke volume refers to the amount of blood that both ventricle will pump, and one single contraction, and that is the key word there. Then we have ventricular diastole, which is the ventricular relaxation. After this is and this again, don't worry about isovolumic ventricular relaxation phase. Is the same thing that I told you before. That's too detailed. But every time you see this isovolumic, that means that all the valves are closed. So what that means is that right after systole, when the heart in the very early stage of relaxation, there is enough pressure that still remains in the ventricles, in the right ventricle and the left ventricle, that do not allow the tricuspid and mitral valve to open, okay? Because this is just at the very end, in the very beginning of the very end of systole and the very beginning of diastole. Also, at this point, there is enough pressure, okay, or drop in pressure in the ventricles that allows the valves, the pulmonic and aortic valves, to close. So at this specific moment, this is when we call isovolumic relaxation. Don't worry about it. You just know because it happens in fractions of seconds, and it's not worth it from the practical perspective. What you need to know is really this one, that at the end, the pressure will drop significantly, right? That will cause the opening of the tricuspid and the mitral valve so the cycle can repeat and the blood can again fill the ventricles. This from the AKG, when the ventricles relax, that generates the T wave, and that represents the repolarization of the ventricles. Very important to know that for the exams. Let's talk about cardiac output. Okay, what is cardiac output? So let's read the concept. It's a measurement of the amount of blood pumped by each ventricle in 1 minute. And I have here in green the key words. So because look, if you read stroke volume, which by the way, how we calculate the cardiac output is by multiplying the stroke volume times the heart rate. The heart rate, as you know, is how many times the heart contracts in a whole minute. And we said that the normal heart rate is between 60 and 100 beats per minute, right? So what's the difference between cardiac output and stroke volume? So stroke volume is the amount of blood that the ventricles pump with one contraction, while cardiac output is the whole amount of blood pumped by the ventricles in a whole minute. So, and remember that in a whole minute, you're going to have a bunch of contractions, and the amount of blood that gets pumped each contraction plus how many times in a minute, that is what's going to determine the cardiac output. So you can see here heart rate, the stroke volume is determined by the end diastolic volume minus the end systolic volume. Don't worry about that. You just know that the average stroke volume is 70 to 80. For your exams, most likely use 70 milliliters of blood. And so if somebody has, for example, heart rate of, let's say, 80, right? And you want to calculate the cardiac output, what you do is you multiply 80 by 70. 70 is the average stroke volume, 80 the uh, the heart rate, and then whatever result is that, you can do it. I'm kind of lazy, I don't want to do it. So whatever result you have there is the amount of blood that the heart has pumped in a whole minute. That is the cardiac output. Don't worry about all of this. This is kind of more detail about the stroke volume that we normally, again, we just said that an average person that weighs 70 will have a 7. That's to keep that simple. Now, if you want to go more in depth and detail about that, like preload is basically the amount of blood that enters into or that is delivered into the ventricles during diastole. Contractibility is how strong the heart squeezes, in this case, the right and left ventricle. And the preload refers to the force that oppose to the stroke volume, to the ejection. So, when the heart contracts, it's pumping out blood, right? Out. So the afterload is the resistance of the arteries, or in this case, that against which the ejection fraction will have to kind of move forward. So that is what we call the afterload. But again, don't worry too much about it. Or if you want to read about this in more detail, you can do it in your books. About the preload, which we know that is the amount of blood that gets delivered during diastole, is there are some things that can increase the preload. As you can see, imagine that if you have a prolonged diastole, that will increase the filling time, or if you have an increased venous return, that will increase the preload. And eventually, if you increase the preload, basically I was telling you, if you put more blood inside the chambers, the ventricles, you're going to have an increase in the stroke volume, which will increase the cardiac output at the end. It's not necessarily super important to get too crazy with those details, at least at this level, right? It's more for the level of medical student, med school. Contractibility is again the force of contractions. And of course, if you have a stimulus that makes your heart to squeeze harder, the stroke volume, of course, will increase, right? Those are examples of chemicals that increase the contractibility. If you look at this, epinephrine makes sense, right? Epinephrine, which is adrenaline, it helps, but then there's other things like hormones and blah, blah, blah. Afterload is what resists, right? A resistance against the ejection. And the higher the afterload basically means more resistant. That decreases the stroke volume, decreases the cardiac output. But when you have a decrease in the afterload, you're going to have a decrease in the stroke volume in this case. All right, so you can even forget about what I just said before. Again, it's just maybe you have a question in your quizzes, but not in your exam. Now, this, you must know that. So I'm going to invest a little bit of time here. Our rate, you must know what's the normal value, and you must know the medical term when you have a heart rate below 60, so bradycardia, and when you have a heart rate above 100, this is what we call tachycardia. And what I want you to mention here is that always, 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 tachycardia at rest, meaning that the person is not doing exercise, or yourself, you're not doing exercise, or you have not been eating or drinking something, Coca-Cola, coffee, or whatever that can increase your heart rate, let's say that at rest, is always abnormal. It's always pathologic. So tachycardia, somebody with a heart rhythm above 100 is always abnormal. Now, bradycardia can be abnormal or can be normal. Remember when I told you that some athletes will have a better big hearts? They're some efficient, so strong that they don't need to contract that much or that fast to perfuse your organs, and they might have heart rates below 60, and that doesn't mean that they're sick. So when you have somebody with a heart rate below 60, what you need to ask if the patient has symptoms associated with that. If the patient has no symptom or is asymptomatic, that probably is normal or just physiologic. Now, if the person has heart rate below 60 and has any of these symptoms, then the person might have some conditions, right? Let's say hypothyroidism might cause that, or people who take medication for blood pressure and then they are taking a dose that is too high, beta blockers they can cause bradycardia, blah, blah, blah. And the same is true for tachycardia. Most of the time, tachycardia, again, is always abnormal. We're going to briefly describe some of these correlation between the cardiac output and the heart rate. Um, and to keep it simple, just remember that the cardiac output is how much blood your heart pumps in a minute. And that is important because that means how much blood your heart delivers to the tissues, right? Or to your organs, how much blood is pumped to your tissues in a minute. What that means is a low cardiac output means low perfusion for your tissues. And high cardiac output means more perfusion, more amount of blood delivered to your tissues. So the concept here is with heart rate, remember that that heart rate is an important part to calculate the cardiac output. You multiply that by the stroke volume. So under normal circumstances, if the heart rate increases or the heart starts beating faster, the cardiac output, of course, will increase. You can just logically think the faster your heart contracts, more blood will be delivered, right? Because the cardiac output is, the heart is just pumping more. Now, in some point, depending on, let's say, for example, if your heart rate is 120, like when you're running, for example, yeah, that will increase your cardiac output. If your heart rate is, look, about 160, the cardiac output remains stable. But then once you pass that heart rate, once you pass 160 beats per minute, see what start happening with the cardiac output. Start decreasing. Now, here's my question for you. Why do you think that happens? Why do you think the cardiac output decreases when you have a heart rate that high? So take a minute, pause, and try to think and see if you can get to the answer. All right, now I'm going to tell you if you didn't pause. The reason why is because the faster the heart contracts, you will have less time to allow the heart to fill the chambers during diastole. So, in other words, you can imagine that you need to relax so the chambers can fill with blood and then get pumped, right? But if your heart is bumping too fast, you're not allowing the heart to have enough time to fill the chambers with blood, and that's why your heart is not pumping enough blood. That's translate to having a decrease in cardiac output. So there is a point in which if your heart rate is too high, the cardiac output will decline because uh, diastole is too short and there's not enough time to let the ventricles fill with blood. This is another important concept that we need to go through is the cardiovascular center. It's something you learn in AMP1. Basically, what it's saying is which part of the brain can control or modify your heart rate, your not just the heart, but also that's what it's called cardiovascular. We're going to take care here of the cardiac part of the heart to increase or decrease, right? So these take place in the brainstem. If you remember, the brainstem has three parts: medulla, the midbrain, the pons, and the medulla oblongata. So that one, medulla oblongata, is the part where you have the respiratory center and the cardiovascular center. And what that means is if we integrate that with what we learn and the nervous system about the sympathetic system and parasympathetic nervous system, remember that those are part of the autonomic meaning the things that we cannot control voluntarily, system. So we can see here that in represented, right? So what happens is that if for some reason your blood pressure, let's say that, let's do that a scenario and talk about homeostasis, how this is important. Let's say that we have receptors, as we mentioned before, one that detect changes in pressure. Those were called baroreceptors. And let's say that we have these receptors that detect that your blood pressure drops. So blood pressure has dropped and your heart rate has dropped, right? That is a problem because low blood pressure, low heart rate, that means low cardiac output, which means not enough blood being delivered to the tissues. So the receptor sends signals to the medulla oblongata saying, hey, we have low blood pressure and low heart rate. How we can fix that? So the medulla oblongata has two options. The medulla oblongata can activate the sympathetic or can activate the parasympathetic. Now, the parasympathetic, notice that what it does is decreases the heart activity. The sympathetic stimulates the heart activity. So in this case, once the medulla oblongata receives that information, the medulla oblongata essentially stimulates, in this case, the sympathetic, which is a group of nerves that travel to the spinal cord and at the level of the thoracic region. These nerves will leave the spinal cord and will travel through the body to reach specific parts of the heart, the most important one here in the SA node, and that will stimulate the increase in heart rate. And if you have an increase in heart rate, that means an increase in cardiac output, which means increase in blood pressure. That is the first response, right? And we can do the same thing if the opposite will happen. If the blood pressure goes up, in this case, you're going to stimulate the parasympathetic nervous system through the vagus nerve, and that will do the opposite. Will decrease heart rate, decrease cardiac output, decrease blood pressure. So what are the big things to remember here? The cardiovascular center, that without these nerves, your heart still will contract because we have the SA node, which is the pacemaker of the heart, is independent of the nerves. But when there are circumstances that require your heart to increase activity or decrease, that's when the nervous system takes place, and the cardiovascular system is located here in the brain, specifically in the medulla oblongata. We're almost done with the heart. Let's review the membranes and layers of the heart. So far we just learned that the heart is this organ that contracts, and we mentioned that it's located in the thoracic cavity and the pericardial cavity. And as I've been saying many times in the MP1, all your organs, they're not naked. They're wrapped by membranes. And because also organs, there are hollow organs, they are covered by membranes from inside. So what I'm saying is we need to learn that the heart, there are parts of the heart that receive specific names, membranes, innermost, middle, and outermost. Okay, so to see that, we need to take a little piece of heart, okay? This, if we magnify this, we know that this is the left ventricle, so I'll put here LV for left ventricle. So this is the inner, and this is the outer, right, part, right? So notice that the inner part, the most deep part or layer of the heart is this very thin endocardium layer. Okay, so the endocardium is what covers the heart valves, the heart chambers, and continues with the endothelium lining of the blood vessels. So what we have is this endocardium is and I'm gonna do it here, maybe you can appreciate it better, is it runs or is the innermost layer that covers all the chambers, all the valves, and even continues through the aorta, and the same thing applies for the superior vena cava, the right atrium. Everything internally has this endocardium. And even though you have a bunch of blood, right, the blood cannot be used to get oxygen and nutrients to feed this important part of the heart called the myocardium. So that's why we need the coronary arteries that supply blood to the heart to feed the cells, the myocardium. Because again, you have this endocardium and it's kind of impermeable. Nothing passes through here. Okay, so which leads me to show you the next part, the most thicker part of the heart, the myocardium. So the myocardium is the functional part, right? It's composed by these cardiac muscle cells. And as I mentioned before, the left ventricle has a thicker myocardium than the right because reasons that we already mentioned about the systemic circulation. So we're learning these layers of the heart from inner to outer, right? So endocardium, the thicker layer of the heart, is the myocardium. This one is the one that confuses everybody. Remember that these organs in the chest and the abdomen, they have this outer layer, and this outer layer has two layers. We call this in AMP1 the serous membrane. And we said, think about the serous membrane as this plastic bag that covers the organ. And I use the analogy of having fruit like a watermelon or whatever, and having two plastic bags to keep that watermelon. So the first plastic bag is the one that is actually touching the watermelon. That's what we call the visceral layer. And the second and outermost layer is what we call the parietal. So in the heart is the same thing. Notice here, the myocardium is this layer. And then we have this one, which is light purple, I guess. I'll call this A. This one here, I'll call this B. And there's a space, I'm going to put S, between them. This space is what we call the pericardial cavity, is the space between those two layers. A, the innermost, the one that is touching the myocardium, is also known as the visceral pericardium or visceral layer. Now that has a name. Specifically, it's called the epicardium. And if you remember that word epi meaning on top of, this is the heart, the cardium, the most important part. So the layer that is on top of that, epicardium, which is the same then visceral pericardium. Then we have the space, which is called the pericardial space, that contains a little bit of fluid called the pericardial fluid. And then after that, we have B, which is the visceral layer of the pericardium. And then there's an extra one called the fibrous pericardium. Don't worry about it. So don't worry about that one. But this is the parietal layer. Now, this fluid between the visceral or epicardium and the parietal pericardium is important because it helps to lubricate. So when the heart contracts, there's not a friction, or reduces the friction between the layers during contraction. Okay, that is the main function of that. Now, there are situations where you can have inflammation of one of these layers, or sometimes cancer, or sometimes after uh, a heart attack, you can have problems in which the fluid in this pericardial space will increase. And that's what we're going to see next. What is the name of that condition when you have an increase of fluid in the pericardial space? So we can see here the normal pericardium. The condition is called pericardial effusion, when you have fluid building up between the pericardial space. Eventually, if the fluid is enough, they will generate pressure and restrict the ability of the heart to relax. Then this condition is called cardiac tamponade. And the cardiac tamponade is an emergency. The cardiac tamponade can be fatal. The reason why is because the cardiac tamponade, you can imagine that as more fluid accumulates and more external pressure gets inside of the heart, that means that, and let's say you're critical thinking, let's see if the answer is here, I don't think so. Which one do you think will be affected by a cardiac tamponade? Option number one, systole. Option number two, diastole. Option number three, both. Option number four, none of them. Okay, so take a minute, take a pause. Which one do you think, if you have a cardiac tamponade and you have all this pressure here, which one do you think will be affected? All right, the main thing that will be affected is diastole, right? Why? Because not systole, because systole is when the heart actually contracts, so not a big deal. But the thing is, diastole is when the heart relaxes, and it's when the heart, when the chambers needs to expand and relax uh, to let the ventricles, um, to fill the ventricles with blood. So if you have a cardiac tamponade, the problem is that you're not allowing the ventricles or the chambers to relax because this external force, and that can cause problems with, with the cardiac output because you will have no blood to pump, and that's why it's fatal. The way how you treat it, because when treated, you can imagine that can lead to death, is by something called pericardial paracentesis, uh, uh, pericardiocentesis, which is basically making a little hole and draining that fluid. And most of the time, when you take that fluid out, then that lets the heart go back to the normal shape. Now, the treatment is out of the scope of your learning objectives. You just know that cardiac tamponade is when this pericardial fluid filled with excessive amount and can lead to problems with relaxation on the heart. To conclude the class, this is just the embryological development of the human heart. Just read about it. Important here is just to mention that the heart is the first organ to form and to function, and is the last one. And what I want to tell you here is that about this rotation that I mentioned before, remember I told you the apex is backwards. Initially, during development, the apex was in the bottom, I mean in the top. But as the heart will turn, then the apex becomes upside down. It's because of this uh, uh, rotation that takes place. Not worth it to memorize all these names. The only reason I have here these pictures to show you that rotation that will take place and will make the apex be in the bottom. That's it. We cover all the content. I hope after this class you have a better understanding about the heart, how the heart works, and I'll see you next time when we cover the vascular part.